Hello everybody, it's Mrs. Pound, and today is the last video for Chapter 3, Introduction to Cells from BJU Biology 4th edition. And today we're going to take a look at Section 3B on cells and their environment. So our objectives today are to compare and contrast passive transport and active transport, distinguish between facilitated diffusion and active transport, and compare and contrast endocytosis and exocytosis. So the section we're going to look at today is 3.5, transportation across the membrane. The first thing we want to talk about is the different types of transport. The first is passive transport. This is the movement of molecules across a membrane with a concentration gradient without the expenditure of chemical energy. And I have a picture of a skier because remember when we talked about diffusion and osmosis, which are types of passive transport, we said they follow a concentration gradient that is downhill. We always go from higher concentration to lower concentration, just like we go from higher elevation to lower elevation on a hill. And so when we do that, we don't use our own energy to go down a hill on skis, okay? And so just like there is a uh, height gradient for a hill with the concentration gradient, as long as you're going with the gradient going downhill in a sense, there's no energy expended. And some examples are diffusion and osmosis that we've talked about before. Now, there is such a thing called facilitated diffusion. This is when a substance is helped across the membrane with the concentration gradient. And that's because not all things can make it through this membrane. So they have to have some holes, like this protein channel, which is a transport protein. So there are things called transport proteins in the cell membrane, and this is a protein molecule embedded in the cell membrane that mediates the passage of certain molecules. So some of these proteins in the cell membrane, and there would be lots more than just one, help facilitate transport. Now, this is still passive transport. They can't go against the concentration gradient in facilitated diffusion because it is diffusion. There is also what's called active transport. This is the use of cellular energy to pump particles across a membrane against the concentration gradient. So this time I have a cyclist going uphill. They're using a lot of energy to go uphill against the gradient, against the concentration gradient as well. It takes energy. And one of those things that requires energy is endocytosis. This is the process a cell uses to transport substances in bulk across the membrane into the cell. And generally, okay, if it's engulfing something, um, it's going against the gradient because it has more nutrients inside of the cell than outside of the cell, so it requires energy. And the first type of endocytosis I want to talk about is called phagocytosis. This is the engulfing process by which cells take in solids by forming vacuoles. So phagocytosis basically is kind of, we call it cellular eating. It's going to engulf. Okay, and an amoeba, you will see this where they will actually extend out and surround the food particle that they are going to eat using phagocytosis. And they actually do engulf that entire food molecule, whatever they are eating. Okay, or perhaps it's a bacteria cell, whatever they are eating, they will totally surround. And so phagocytosis is cellular eating. There's also pinocytosis. This is the process by which fluid enters a cell in bulk by being surrounded and engulfed by the cell membrane. So again, it will engulf uh, some fluid. Okay, a lot of times pinocytosis is called cellular drinking. Okay, cellular drinking. is pinocytosis. And then there is exocytosis. And this is the process in which vacuoles or vesicles fuse with the cell membrane to release particles or substances from the cell, things like hormones, enzymes, and waste. These are generally things that are too big to go across the cell membrane, but exocytosis, exiting, exterior, outside. Endocytosis, we're trying to get things in. Endoskeleton is a skeleton that is inside something. So endo in, exo out. Okay, exo exiting. That's why I have an exit sign in the background. So our objectives today were to compare and contrast passive transport and active transport. Remember, passive, no energy, active requires energy. Distinguish between facilitated diffusion and active transport. Facilitated diffusion still with the concentration gradient, just helped a little bit. Requires no energy, active transport requiring energy. And to compare and contrast endocytosis and exocytosis. Endo going into the cell, exo going out, exiting. Okay, so don't forget your 5 questions in the margin and continue watching this video to answer the questions at the end.